Oh.
All right, here they come.
Do we get to see them?
No.
Ah, bummer.
All right, so we're going to get started in a moment. For those of you that have just joined, we'll let a few more people trickle in, and then we'll start with housekeeping items for everyone to learn the tools on this Zoom webinar. Okay, let's go ahead and get started. I just wanted to let everyone know before we begin, thank you for joining the SSR Exotic Species webinar series. This first webinar is from worm to germ, germ cell development and regeneration in planarians. And we've got the speaker, Dr. Newmark, and the moderator, Erica, Dr. Matunis, Erica Matunis. I wanted to let you know that you are all muted as attendees. So the main way that you'll be interacting with the speakers today is through the Q&A box that you can find at the bottom of your screen. Please go ahead and enter the questions there, and they will be moderated by Erica Matunis. And the presentation will be 30 to 40 minutes long, followed by a Q&A session. So please keep in mind that your questions may or may not get answered until we get to that point during the Q&A session. If you need help for any reason, please feel free to email us at administrator@ssr.org or meetings@ssr.org. And with that, I will hand it over to our moderator.
Thank you very much, Simon, for this opportunity, and thanks to SSR for putting on this great webinar. So my name is Erica Matunis, and I see a few people that I recognize already in the participant list. Thank you so much for being here on this most distracting day. I am a professor of cell biology at Johns Hopkins University in Baltimore. And I'm a proud SSR member. My lab works primarily on fly germ cells, and we study the testis niche, spermatogonial stem cell niche. And we also do a little bit of mouse work. And I think you've seen all the standard things about my bio in the blurb that announced this. So I can give you the personal version. I went to a small liberal arts college in Washington State, and I grew up there. I went to graduate school in Chicago, and then I kept moving east. I ended up doing a postdoc at Rockefeller with Steve DiNardo, where I learned fly genetics. And then after that, I went to the Carnegie Institution of Washington, and that's here in Baltimore, where I am now. And that is where I met Phil at Carnegie. And that was a wonderful time in our lives. So since then, I moved on, and I have my position at Hopkins across town. But Phil, I will say something about now. We are so glad to have him presenting his lab's work today. His position now is an investigator at the Morgridge Institute in Madison, Wisconsin, and that's a small institute like the Carnegie. And he does amazing work on not just planarians, but schistosomes and other kinds of tapeworms, parasitic worms. And I would say that you guys have probably, many of you have probably heard some of his beautiful presentations. He's an excellent communicator. He's also a gracious teacher. He's just taught an enormous class that he wasn't required to do. He's a wonderful, wonderful colleague, the kind of person that you will always go to with a question, and he's very thoughtful and insightful. And he's an amazing mentor, and he's a Howard Hughes investigator. He continues to use cutting edge technology to push the field that he's in forward. And it's been such a pleasure to see this all unfold, because he grew up in Cleveland, and he got his BA in biology in 1987 at BU. And then he went to Boulder, and he did a postdoc there in a fly lab and studied fly germplasm with Bob Boswell. And after that, he was just very brave. He did something no one was doing. He went to Barcelona for a postdoc to study flatworms with the Begunia lab. And he was there until 1996. And then he actually did another postdoc at the Carnegie, where he brought worms and worked with Alejandro Sanchez Alvarado to basically establish or reestablish the system as as a way to study regeneration with modern tools, because right around that time, of course, RNAi was being discovered downstairs. So it's all gone amazingly well since then. After he left the Carnegie, he got his first faculty position at UIUC, and he moved up through the ranks there. And then he's been at the Morgridge since 2016. And I think you're all in for a wonderful, wonderful talk. So thank you, Phil.
Okay, make sure I'm unmuted, and I'm sure someone will let me know if you can't see whatever it is I'm doing here. So our pointer's working, we're all good. Okay, so to start, I want to thank Erica for the gracious introduction. It's really a pleasure to get to do this with her, and also to thank the folks at SSR for inviting me and giving me this opportunity to share some of our work and talk about these exotic creatures that I work on. So today we're going to be talking about these flatworms known as planarians, shown here, and we'll walk you through a little bit about their biology. So what I'm going to do today is I'm going to assume that most of you are very much unfamiliar with planarian biology and especially their reproductive biology. So I'll start by giving you some background, just so you know some of the things that we've been able to do, some of the things we're learning. And then I'm going to spend the majority of the time talking about unpublished work to identify genes that are required for female germ cell regeneration in planarians. And then I'll move on to show how that work has led us to uncover a really fascinating role for monoamine transmitters in regulating germ cell development. And the source of these transmitters seems to be the somatic gonad. So I'll walk you through that work as we go. All right, so to begin, we'll start with probably what most people are familiar with planarians is their amazing ability to regenerate. So if one lops off a planarian's head, within a week or so, it's capable of growing back entirely. And this amazing process of regeneration is driven by stem cells that reside in the adult planarian's tissues. And these stem cells respond to the wound by proliferating and migrating towards the wound surface. And in the course of several days, they form this unpigmented structure known as the regeneration blastema, in which the missing tissues will be reformed. Okay, so within about a week or so, this animal is capable of regenerating its photoreceptors, its chemoreceptors, rebuilding its brain, rewiring it, and it turns into a perfectly functional little planarian. Now, again, these regenerative abilities are due to this population of stem cells that these animals maintain throughout their adult lives. Flatworms basically have just a single proliferating population of somatic cells. And these are undifferentiated cells known as neoblasts. So if one gives a nucleotide analog to a planarian, what one finds is there's this dramatic proliferation throughout the worm in only these undifferentiated cells. And these cells are, as you can see, are scattered throughout the worm's body. And these are the cells that will respond to amputation to produce the missing tissues. Now, work from several labs over the years has shown that these neoblasts express many genes that are associated with germ cell development in other organisms. So neoblasts express genes like pewees and VASAs and tutor-like genes, just to provide a few examples. So there's a really interesting aspect of these neoblasts that sort of relates them a little bit to germ cells and the pluripotency program of germ cells. Now, work from several labs has also shown that this population is quite heterogeneous. So amongst this large population of proliferating cells in the worm are cells that have already been committed to various differentiated lineages. And we know that based on their expression of various tissue-specific markers. And most importantly, we know that within this population of cells, a subset of these cells are truly pluripotent. And that's been shown by beautiful single-cell transplant experiments from Peter Radine's laboratory at the Whitehead Institute, in which Peter and his students were able to take individual neoblasts, transplant them into lethally irradiated worms, and restore viability and regenerative abilities, showing that these cells are capable of giving rise to all cell types in the worm. All right, so in addition to this amazing ability to regenerate a brain, those of you in SSR will also really appreciate that these animals can also regenerate their reproductive organs. And the experiment I'm showing is one that was actually performed by Thomas Hunt Morgan back in 1902. So what Morgan did was he took a sexually reproducing planarian, and I'll tell you about the anatomy in a little while, but basically he could take the head of this planarian and remove it from all of the reproductive tissue, which is down here. When he did that, he then makes a head fragment that completely lacks all reproductive organs and germ cells. And of course, this little piece will regenerate a tail, remodel, and ultimately give rise to a perfectly proportioned little planarian. Now, as this animal feeds and grows, it will basically completely rebuild all of its reproductive system post-embryonically. And from this very simple experiment, Morgan recognized that these animals are capable of making their germ cells de novo from their somatic tissues. And this is really fascinating because this is really quite different than the way we think of the way our favorite model organisms make their germ cells. So in organisms like Drosophila and C. elegans, which have told us a huge amount about how germ cell development works, those germ cells are specified by localized determinants that are basically built into the egg. And the cells that inherit that special cytoplasm, the germplasm, will go on to make germ cells. But here, the planarian is actually able to specify its germ cells based on signaling from surrounding tissues in a way akin to how germ cells are specified inductively in mammalian embryos. Okay, so this is a really interesting question. How is it that these somatic stem cells are capable of giving rise to germ cells in these creatures? Now, I want to tell you a little bit about the reproductive biology, since this is a seminar for SSR. Planarians can actually reproduce in two different ways. So several species of planarians can reproduce asexually. And what these animals will do is essentially adhere their tail to a substrate, their head will pull away and stretch, and the animal, I guess you may be able to see my hands, but essentially the animal will kind of stretch and stretch a bit like a rubber band, and eventually it will result in fission, usually posterior to the pharynx, creating a tail fragment that will make a new head, and then a head fragment that will regenerate a new tail. And so the asexual strains of these organisms use regeneration as their mode of reproduction and allow them to reproduce asexually. Now, it turns out, as I alluded to before, there are also sexually reproducing planarians. These are simultaneous hermaphrodites. And so what's cartooned here is, so here are the photoreceptors. Behind that is the upside down U-shaped brain, the cephalic ganglia. And at the base of the brain, as you'll see in a little while, are a pair of ovaries. Dorsolaterally are scattered dozens and dozens of individual testis lobes. Okay, and then there's a complex copulatory apparatus that these hermaphrodites use to actually mate and deposit sperm. They're non-self-fertile. The complicated plumbing that the animals use for moving the gametes around actually prevents self-fertilization. So all of this this amazingly complicated reproductive stuff is built post-embryonically after the animals hatch out of the egg. Now, many species of planarians are capable of actually switching between asexual and sexual reproduction, depending on poorly understood environmental cues. For example, there's some seasonal component, but very little is understood about how that works in those species. But the species that we study, the one that we've developed as a model organism called Schmittea mediterranea, for which we've developed genome sequence and many, many of the tools that you'll hear about, it turns out Schmittea mediterranea is not able to interconvert like this. There are specific strains that reproduce either asexually or sexually. These strains are geographically isolated, and the asexuals the asexual strain contains a chromosomal translocation that is lacking in the sexual sexual biotype. Okay, so when I began my lab at Illinois back in 2001, we hoped to exploit this difference between the asexual and sexual modes of reproduction to understand more about germ cell biology and how these animals were regulating the development of germ cells. And one of the first things we did was to, in a set of proof of principle experiments, was to characterize the conserved germ cell regulator nanos that's associated with germ cell development in every animal that's been studied. And to our great surprise, it turned out that when we looked in the asexual strain of Planarian, we were able to see nanos expressing cells in the primordia or in the regions of the worm where the gonadal primordia would form. So in fact, these asexual Planarians that lack all these accessory reproductive organs actually have primordial gonads. They have both primordial testes and, as they get bigger, primordial ovaries as well, all of which express nanos. So to our great surprise, in these asexual Planarians, they can specify their germ cell lineage, but these germ cells are unable to develop further. Okay, now I'm going to be talking to you today mostly about oogenesis and how the ovaries are, how the female germ cells develop in these worms. But I do want to tell you a little bit about testis structure as well, because you will see this a little bit later in the talk. So I wanted to give you a sense of what this looks like. So what you're looking at here is just an animal stained by fluorescence in situ hybridization to detect a late male germ cell marker in green. And so you can see the testes dorsolaterally, just how abundant they are in these worms. And now if we zoom in a single confocal section of a whole mount worm in which we're now detecting nanos mRNA in magenta, what you can see, I hope, is that these individual testis lobes are arranged very much like a mammalian testis in that there's an outer spermatogonial layer and differentiation happens towards the lumen. And the nanos positive cells that I showed you in the asexuals as just tiny clusters of cells in primordial testes, these nanos positive cells represent just a subset of the spermatogonial outer layer. And we know that nanos is required for the continuing development of these cells because when you knock down nanos, you completely lose the testes. Now, if we dissociate the Planarians and now remove and just stain, use nuclear dyes to look at the clusters of their developing sperm, what we can see are clusters of spermatogonia that will undergo 3 rounds of division with incomplete cytokinesis to make 8 spermatocytes, which will then undergo meiosis to generate 32 interconnected spermatids. These then undergo a really beautiful process of spermiogenesis in which the nuclei elongate, and then these elongated nuclei also, or the developing sperm, also build 2 9+1 flagella, which stick out of the anterior end or the top end of the sperm. So these are really kind of wacky sperm. But the point here is that just by looking in whole mount organisms, we can see all the different stages of spermatogenesis. And by using RNA interference, which we can do very effectively in these animals by feeding them double-stranded RNA, we can actually identify genes that block all of these various different pathways in spermatogenesis. And so over the years, what our lab has been able to learn a lot about spermatogenesis in these worms and male germ cell development simply because the animal biases us to do that because of the abundance of testis tissue. And so in fact, over the years, there's been a real paucity of understanding of what's going on with the female germ line. Oh, and I guess I had one last point to make here, which is here. So one of the things that came up, which will be of interest to this audience, is that the role of a conserved DMRT homolog in male reproductive development. This DMRT or DM domain containing gene, DM domain encoding gene, is actually expressed in all of the male reproductive organs, in the somatic tissues of the testes, as well as in all the male accessory reproductive organs. And knockdown of this gene leads to loss of male germ cell specification and loss of all the male reproductive organs. So anyway, so that's a little bit about what we've learned about males so far, but we really wanted to also understand what was happening with female germ cells. And this is where Umair Khan has come in. Umair is a senior graduate student in the lab. Here he is with our lab mascot, Schmitty, and one of lab mate and his classmate, Jarong Gao, who actually just finished her PhD. Jarong works on schistosomes and rotifers, and a project I'm not going to have time to talk to you about today. For those of you who are wondering, so our lab mascot Schmitty is not a poop emoji. This is a planarian, but our office administrator did not understand why we were posing with poop emojis for our pictures. Anyway, so I'm going to talk to you about Umair's work to understand how these animals make their female germ cells. Okay, and again, this is a really important question. There are related species of planarians that are parthenogenetic. And so understanding how those pluripotency or totipotency programs are built into these eggs is really, really of interest. And so there's really important reasons for understanding how these animals can regenerate their female germ cell lineage. Okay, so I've already introduced you to the anatomy with the brain, the ovaries at the base of the brain, dorsolateral testes, and then here you can see the ducts, the oviducts up which the sperm must swim to fertilize the eggs as they are stored in the tuba, which is a structure right outside the ovary. And again, all of these ducts are built post-embryonically. So if we zoom in now on just the DAPI cross-section of a fluorescent C2 stained ovary to detect early germ cells. This was the marker nanos. What you can see here are the early germ cells kind of at the periphery here, and you can see these oocytes ready to exit into this sperm storage organ down here. Now, those of you that work on vertebrates are used to being able to actually dissect out organs to get organ-specific transcriptomes. We can't do that with planarians. Planarians are acoelomates, which means there's no extra space in the tissue. So all of these organs are kind of glued together by a mesenchyme without any extracellular space. So essentially, trying to dissect out any given ovary or any given organ is kind of like trying to dissect wet toilet paper. So that doesn't work very well. So what Umair has done to compensate for the male specific bias that we get when we look at whole organism RNA preps, et cetera. He's worked with a former postdoc in the lab, David Forstoffel, to develop laser capture microdissection in planarians, where now what Umair can do is take cross sections through the animal and now identify in a microscope ovaries, testes, or non-gonadal tissue, and basically remove them using one of these laser microcapture instruments, and now generate through RNA sequencing of the isolated ovaries, testes, or non-gonadal tissues, generate transcriptomes from each of these tissues. So Umair has done that, and he's been able to identify really thousands of candidate genes that are expressed in sex-specific manner. For our purposes today, we're going to focus on the ovary-enriched transcripts, of which he's identified roughly 1,600 or so. So this just shows that he can isolate RNA from these tissues, and he can detect differential gene expression, for example, to find ovary-enriched versus a whole collection of almost 4,000 testis-enriched genes, and then other genes that are basically show no sex-specific bias. But of course, what we really want to do is now not just identify these in this manner, but really to validate them. And we can do that by whole out in situ hybridization, which we can do in relatively high throughput in these animals. So what I'm showing you here are individual colorimetric in situ hybridizations for a panel of genes that came from Umair's ovary-enriched list of genes, of candidate genes. And the main point to take away from here is that, in fact, he is able to see ovary-enriched expression of many of these genes. And intriguingly, he sees many, many different kinds of patterns. And we'll walk through why he sees such differences in patterns. But for our purposes now, I just want to say this worked really, really well. 90% of the genes that he's tested by in situ hybridization are show enriched expression in the ovary. And we believe the ones that don't show enriched expression are expressed below the detection level. So he's just not able to see them. So he did a really good job with his laser microdissection, and these transcriptomes really are a really good source of identifying genes that are expressed specifically in the different gonads. All right. Now, so this now is just, I'm going to show you a couple of genes that we'll be looking at throughout the rest of the talk. KLF4 is, of course, this conserved pluripotency factor. It turns out to be expressed at the earliest stages of germ cell development in both the female germ cells, as well as, and I'll show you this later, in the male germ cells. And in the female germ cells are actually detected in a field at the base of the brain. And so what you're seeing here, the DAPI staining here, is actually reflecting the the density of nuclei of the Planarian cephalic ganglia. And you can see the ovaries here, where the oocytes are labeled by this oocyte-specific marker, LECG. And then you can see the KLF4 positive cells in these streams outside the ovary. So by performing fluorescence in situ hybridization with these various markers, Umair has been able to define genes that are expressed at many different stages of female germ cell development. What he's done here in this overlay is combine the KLF4, which labels the earliest female germ cells, and LECG, which labels oocytes. So he's pooled them into a single channel, and then is showing you here the expression of a gene PKS1, which seems to be expressed throughout the development of the female germ cells. Here's a gene, an initiation factor, a translation initiation factor that's expressed only in mature oocytes. So you only see them in the mature oocytes that are basically at the point of exiting the ovary. And so in this manner, Umair has been able to define gene expression changes that lead from the neoblast, the stem cells which express PWE, as I alluded to earlier, and then a germinal histone H4 as well, to the early stages of germ cell development that express this KLF4 gene and the nanos gene that I mentioned before. PKS1 seems to label throughout the stages of oocyte development, and then genes that now become expressed only in later stages of oocyte development, like LECG and then this initiation factor. Okay, now of course you all know that the somatic component of the gonad is extremely important, and the somatic cells play an important role in regulating the germ cells. And Umair has been able to identify from his transcriptomes and subsequent in situ validation several genes that are expressed within the somatic cells of the ovary. I'm showing you here a FOXL homolog, which I'll talk to you about in a minute. So again, he's pooled the germ cell markers in magenta. The putative somatic markers are now labeled in yellow here. And the overlay, you can see that basically these FOXL is labeling a different cell population than the germ cells. And you can see how it basically is wrapping around the oocytes. And here's a delta homolog that also is not expressed in germ cells. And then the endothelium converting enzyme that is also expressed in a subset of these somatic cells within the ovaries. Okay, so I've just told you that we can isolate RNAs in a tissue-specific manner, generate transcriptomes, and validate those genes. Of course, what we're really interested in is testing whether or not those genes functional role in the regeneration of the ovary. And so this is how Umair has proceeded with this work. As I alluded to, we can basically just feed the Planarians in vitro synthesized double-stranded RNA in their food. They take it up, and the systemic RNAi works really, really well to knock down gene expression. So he can feed animals over the course of a couple weeks. And then what he does is he amputates the head and the ovaries. So as you can imagine, the head is going to grow back. And as I told you, the ovaries will also regenerate. And so once the head has regenerated and the animals are capable of feeding, Umair can then resume the double-stranded RNA feeding over the course of about a month. And now he can look at the final stage of regeneration and use some of these in situ markers I just showed you to analyze phenotypes to identify functional roles for these genes. And so I'm going to tell you about 3 different interesting phenotypes that have emerged, or genes that have emerged from these studies. One gene that seems to be expressed in the early female germ cells and is required in a female-specific manner. And then I'll talk to you about 2 genes that are expressed in the somatic gonad that also play roles in the regeneration and development of the female germ cells. Okay, so this gene ZFS1 encodes a zinc finger predicted RNA binding protein of the C3H1 class. And that gene is expressed in the field of early female germ cells outside of the ovary, as well as in intermediate stages of oocyte development. And I'm going to, in the interest of time, I'm not going to belabor this, but basically Umair has done double fluorescence in situ hybridization with these various markers to show that a subset of the KLF4 positive early germ cells are positive for this gene, as are a subset of the LECG oocytes. And so this ZFS1 gene appears to be expressed during an intermediate period between the earliest stages of female germ cell development and ending expression before the final oocytes are made. Now, again, this is now showing you the RNAi experiments. The control animals are shown at the top. And what we're doing is now zooming in on the region of the two ovaries in between the brain lobes, using KLF4 and C2s in magenta and LECG to see oocytes in green. And what you can see in the controls is that you can see fully mature oocytes in the ovaries, as well as this field of early KLF4 positive germ cells. But now when Numair looks at ZFS1 knockdown animals, you see this dramatic reduction in the early KLF4 positive germ cells and this loss of oocytes. And this is just quantified here. So again, dramatic reduction in the early KLF4 positive germ cells and a complete loss of oocytes. Now, interestingly, if we now look with one of these somatic gonadal markers, again, here's our control, and you can see the various stages of oocyte development here, and you see delta expression within the somatic gonadal cells. Following knockdown of ZFS1, the germ cells are lost, but the somatic gonadal cells have expanded to take their place and actually build a roughly normal-sized ovary that's just lacking germ cells, which is a really interesting phenotype. All right, so and I should say that this effect is female specific. We see no effect on male on testes development in these knockdown animals. So essentially, this gene is expressed within the early female germ cells, and it seems to be an intrinsic regulator that's required either for the specification or maintenance of these early female germ cells. And then I also showed you that there's this interesting effect on the somatic support cells themselves, suggesting that perhaps the female germ cells are actually signaling bidirectionally. Not only are they getting needed cues from the somatic support cells that are required for their proper development, but it seems like the germ cells are signaling back to them to also control their numbers. All right. So I'll tell you about one of these somatically expressed genes now, FOXL. Again, so here it is in magenta, expressed in a different pattern than the germ cells, which are labeled in green. This is just zooming in to show you that. Again, these FOXL positive cells surrounding the oocytes. And this projection just kind of shows you this more clearly. You see these FOXL positive cells that are just wrapping around the oocytes in green here. And so again, in our control animals, after the heads have been lopped off and the ovaries grow back, the field of early KLF4 positive germ cells is reestablished, and the ovaries look normal. In these FOXL knockdown animals, however, the KLF4 field of early germ cells seems okay, but now there's a dramatic reduction in oocyte development, which is quantified here. So there's really no effect on these early KLF4 positive germ cells, but there's a dramatic reduction in oocyte development in the FOXL knockdown animals. Okay, so FOXL, as you all probably are aware, FOXes are fork head domain encoding transcription factors. What I've told you is that this FOXL gene in Planarians is expressed in the somatic cells of the ovary and is required for proper development of the oocytes. Now, for this audience who know this way better than I do, of course, FOXL2 is the critical regulator of granulosa cells in the mammalian ovary, and is plays an important role in actually maintaining female fate within the ovary. Now, FOXL2 has been thought to be a vertebrate innovation because a role for FOXL genes in the somatic ovary has not been shown in C. elegans or Drosophila. Again, our favorite model organisms that have told us a huge amount, almost, have really defined germline stem cell niches for us. So in fact, the thought has been that vertebrates, the role of FOXL in the somatic gonad here in the ovary was a vertebrate innovation, but because these genes were not seen playing such roles in C. elegans. And it turns out Drosophila and C. elegans belong to the same lineage of invertebrates, the so-called ecdysozoans or molting animals, whereas Planarians fall into a completely different evolutionary branch of the invertebrate lineage. And what I've just told you is that Planarians are also using a FOXL homologue in their somatic gonadal cells to regulate female germ cells. So this is a really, I think, a good example of how looking broadly across the phylogenetic tree can point out potential deep conservation that may be missed by focusing on just a few organisms. Okay. And so the last little piece of this story that I'll tell you about is another somatically expressed gene that we find to be particularly fascinating. So from Umair's transcriptomes, he found that this gene AADC, which I'll tell you about in a minute, he found it expressed in the gonads. Looking ventrally, you guys are now experts. You can see the expression in the ovary here at the base of the brain. And if we flip the animals over dorsally, you can also appreciate that there's actually expression in the Planarian pigment eye cups. Or ventrally, I should say, all these little dots here are expression in neurons of the brain and peripheral nervous system. And then dorsally, you can see expression throughout the testes, actually in the somatic gonad of the testes. And so if we zoom in here, this is just showing you this even better in these colorimetric whole mounts, just a subset of cells in the ovary that are basically forming a mesh around the germ cells, which I'll show you more closely. And again, these cells around the periphery of the testes where we know the somatic gonadal cells to be. Umair has generated antibodies against AADC, and now looking in the ovary here, you can see beautiful expression of AADC protein within these cells that are wrapping around the oocytes, so within the somatic cells of the ovary. All right, so AADC is an aromatic L-amino acid decarboxylase, and we primarily think of this enzyme for its role in regulating the synthesis of monoamine neurotransmitters. So for example, here in the synthesis of serotonin, which starts with tryptophan, and then which is converted into 5-hydroxytryptophan via the action of tryptophan hydroxylase, what AADC does is decarboxylates 5-hydroxytryptophan to turn it into 5-hydroxytryptamine, which is serotonin. Similarly, AADC can convert phenylalanine to phenethylamine, tyrosine to tyramine, and probably most familiar to everybody, DOPA to dopamine. Okay, so why are we talking about this? So it turns out that this generated some really intriguing phenotypes. So as I've shown you in the other cases, the control animals, right, regenerate their ovaries just fine, this full field of KLF4 positive early germ cells, mature oocytes. In the AADC knockdown animals, however, what you can see here is a dramatic reduction of these early female germ cells and a complete loss of oocytes, again, which is quantified here. Okay, so loss of female germ cells. Now remember, these animals are hermaphrodites, so we can just flip them over and look at what's going on with the testes. And so what I'm showing you here are the testes in the control animals. Using this KLF4 marker in magenta, you should just see tiny, tiny dots associated with these testes lobes. Now, by standing with DAPI here and zooming out on the animal, just based on the condensation of chromatin in the spermatid and sperm, you can see the testes beautifully just in whole mount animals. Now, before I show you this phenotype, the knockdown phenotype, I need to do a bit of a digression to tell you about the role of KLF4 in males. So I'm gonna do that now. So it turns out this KLF4 gene, which I told you is expressed in early female germ cells, is also our earliest marker for, is a marker for early male germ cells as well. And so in the experiments I'm showing you here, Melanie Isigonis, a senior postdoc in the lab, has characterized the expression of KLF4 throughout post-embryonic development in newly hatched animals, juveniles as their testes are beginning to grow, and then in fully mature adults. And what you're looking at here is KLF4 expression by fish in green, nanos expression in magenta, and the nuclei are stained gray. And so what Melanie has seen is in these earliest stages in animals as they're hatching out of the egg, in these developing primordial testes where there's just individual or clusters of one or two germ cells, basically all of the nanos positive cells are KLF4 positive. But now as these juveniles begin to grow and their testes begin to mature and they begin to enter the spermatogonial fate, Now only about a third of the Nanos positive cells are KLF4 positive. And by the time she looks at adults, only about 10% of the Nanos positive cells out in the periphery of the testis also express KLF4. Okay, so this is marking really what appears to be the earliest stages of germ cell development in these animals. Now, functionally, what Melanie did to examine the role of the gene, again, is to knock down expression by RNAi. And then she performed this experiment that Morgan did earlier. And she basically took this head fragment that's lacking germ cells and allowed it to regenerate. And in control animals, if one looks where the testes are going to form with using Nanos as a probe, you find these clusters of Nanos positive cells that will regenerate. In the KLF4 knockdown animals, however, there are no Nanos positive cells detected. These animals fail to make their germ cell lineage. Okay, so KLF4 is labeling the earliest stages of germ cell development and is required for their specification. All right, close parenthesis, coming back to these knockdown animals. This is what Umair saw, this remarkable phenotype. We've never seen anything like this in Planarians before. So in AADC knockdown animals, what you see is this hyperplasia of these early KLF4 positive germ cells. So now instead of just seeing a handful of dots, a handful of cells per testis lobe, basically the testis lobes consist almost exclusively of these early KLF4 positive cells. And if we zoom in and look at this again, so here's our control testes with this subset of KLF4 positive cells in the spermatogonial layer. You can see all the different stages of spermatogenesis here as the sperm develop towards the lumen. In the AADC knockdown animals now, what you see are testes lobes that are basically just full of these early KLF4 positive germ cells. And I think this is quantified here. So this is a really dramatic effect. All right. So what I've told you is that by knocking down this enzyme involved in the synthesis of monoamine neurotransmitters, we see different effects on female versus male germ cells. So we see a requirement for AADC activity for generating the female germ cell lineages. So those cells are lost when AADC is inhibited. However, in the male gonad, we see an opposite effect where we see an overproduction or an overaccumulation, I should say, of early male germ cells. So of course, now we really want to understand what's going on here. And this is where I'll just leave you with sort of some of the ongoing questions. Which monoamine transmitters are involved? I told you this is involved in making many, many different neurotransmitters. Which, is it a single one that's involved? Are different ones responsible for these different effects on males and females? Are they acting neurally or are they acting in the somatic gonad as their expression would suggest? So to give you a hint that we think this expression is coming from the somatic gonad, I'm just going to show you some analysis of the existing transcriptome. So if we look at just the serotonin pathway as just a representative monoamine transmitter, so I told you tryptophan is converted to hydroxytryptophan via tryptophan hydroxylase. AADC acts to make it into serotonin. Of course, there are serotonin transporters that will take up serotonin from outside the cell and bring it back into the cells. And there's serotonin receptors that are involved in responding to serotonin signals. And so what Umair has done is used his transcriptomes to ask, can he see evidence of differential expression for any of these components in the gonads? And he in fact does. So he sees expression of one of the TPH homologs in both testes and ovaries. AADC is dramatically upregulated in the ovary. And then he sees differential expression of various serotonin receptors and various serotonin transporters that are expressed differentially between the testes and the ovary. All right, so there's at least transcriptional suggestion that these that the gonads can be responding differently to various monoamine transmitters. So what we're now doing is really marching through trying to ask if we can, through gene expression analysis and in situ hybridization and RNAi, find any of these signaling pathway members that can recapitulate the phenotypes we're seeing, and working with our collaborators at Illinois to actually do metabolomic analyses of the knockdown animals to ask which transmitters are affected, and to then ask if we can rescue any of these phenotypes via injecting these monoamines. All right, so to briefly recap, so I've just told you about several different genes that we've identified through gonadal specific transcriptomes, an intrinsic regulator ZFS that seems to be required for the specification and maintenance of early female germ cells. I told you about FOXL, which acts in the somatic support cells and is required for proper oocyte development. And I told you about another gene expressed in the somatic gonad that acts differently upon female germ cells and male germ cells. So hopefully what you can take away from this is that planarians are a pretty fascinating model where we can use their ability to regenerate to learn some new biology that will help us understand how germ cells can be regenerated. FOXL, I think, is a really interesting proof of principle to say that we can find deeply conserved regulators that are shared between planarians and mammals, sort of validating this kind of approach and this use of this organism. And that this discovery of the AADC phenotypes is also, I think, really valuable because it shows that by looking in unexpected places, you can find unexpected roles for deeply conserved molecules. So these monoamine transmitters, we think of them simply as neurotransmitters, but they're ancient signaling molecules that predate nervous systems. And so there could well be roles for these types of molecules in soma germline communication across animals that have been hard to see because of the systemic effects of perturbing their expression in many different organisms. And just more generally, I think I would hopefully advocate that the kind of exotic creatures you're going to hear about in this series, these unique biological features really do let you uncover some really fascinating biology. And so the last thing I'll say is, why else should we care about planarians? Well, planarians are actually free-living relatives of some really devastating parasites, the flukes and the tapeworms. Here I'm showing you male and female pair of schistosome mansoni, which is a blood fluke that infects, over 200 million people around the world. These parasites have very complex life cycles, often involving at least 2 different hosts, which really does make them a little bit harder to study. And so what we've been able to do is take our knowledge about planarians and really apply it to study their parasitic cousins and really make some, I think, some important progress in understanding some aspects of their biology. So for example, within schistosomes, we found that the adult parasites have stem cells very much like planarian neoblasts. And these stem cells are present throughout the life cycle, driving the development inside the intramolluscan host, which is shown here. So this is a part of the life cycle that develops in its snail host. And even these stem cells are packaged into the stage of the life cycle that infects us. And these stem cells then are triggered to proliferate and differentiate upon penetrating our skin. And then the other group of parasitic flatworms, of course, are the tapeworms. And actually, the tapeworms are basically just planarians on steroids. Every single one of those tapeworm segments is a hermaphroditic reproductive system that's generated throughout the life of the animal. It's basically it lives its life by regenerating constantly. And Tanya Rosario from the lab has just left to start her own lab at Georgia. This beautiful images from Tanya's work to understand the role of stem cells in tapeworm regeneration. So with that, I'm going to stop. I want to acknowledge Umair for his great work on the female germ cell regeneration in planarians. I also want to acknowledge Melanie, whose work on KLF I described. She also basically keeps the lab running and is an incredible colleague. Shout out also to Tanya, whose work I showed at the very end, who's just left for UGA, and also to colleagues who started the schistosome work in the lab, Jim Collins, who's now at UT Southwestern, and then Bo Wong. I generated one of the images I showed about the intramolluscan development. All right, and this is what the lab actually looks like now. I'm sure you're all familiar with the Zoom lab meetings by Zoom, which we're all very much looking forward to not having to do anymore. So I'm happy to stop and take questions. So thanks for your attention.
Thanks, Phil. That was terrific. Oh, we've already got a question. I think Gary is asking, but he's asking it in the chat. And I can see that, but I don't know if, let's see, if you can try to ask your questions in the Q&A, that would be amazing. But I will relay Gary's question. Sorry if I missed, but do you see serotonin receptors in the germ cells or the gonad? And could the AADC also make dopamine?
Great questions, Gary. So at this point, what we know about the, all we know is that from our gonad-specific transcriptomes, we see differential expression of receptors in the gonads. We don't yet know which cell types they're expressed in. So that's part of all of our downstream analysis now, is to ask which cells are expressing these receptors. So we need to now go from global transcriptome differential expression to, careful FISH analysis to see which cells are expressing these things. So that's all ongoing work now. Can we detect serotonin and dopamine and all these things also within the somatic gonad as well? So that's all very much ongoing. Was there a second part of the question?
There might have been. He was asking if the same questions for receptors for dopamine.
Yes, so all that work is now ongoing. So hopefully in a few months, I'll have a lot more to say about that now that everyone's back in the lab.
Jan Sun is asking, first of all, thanks for the great presentation. He's wondering whether female germ cells also divide to form syncytial clusters. I was wondering the same thing. And a follow-up to that would be whether each oocyte is wrapped by somatic cells and how they get released into the tuba.
Yeah, so I don't think we have evidence for cysts of female germ cells in these guys. That said, we really need to go back and look carefully by EM now, because a lot of so at the moment, there's not good evidence for, but I would also say that it's not by any means conclusive. And I think we just need to look a lot more carefully. Now that we have these antibodies in hand, like AADC, which is really staining those somatic gonadal cells really well, we can really look carefully. They do seem to be wrapped. The germ cells are clearly wrapped by the somatic cells. Whether they're being escorted by clusters of somatic cells as they work their way through, exactly how the somatic gonadal cells are turning over, we don't have a clue whether the cells are being passed off between them or if there's a constant flow through. So that's all ongoing work. And how the tuba basically kind of opens up to allow these oocytes out. And how that works, we're totally unclear. But basically that's where the sperm is stored. And yeah, I don't have anything intelligent to tell you about the mechanisms of release from the ovary yet, unfortunately.
Great. Eulina Volkan-Filas says, fabulous talk. I noticed that in AACD mutant hypoplastic testes, that the germ cells aren't progressing through meiosis. Do you think they may be behaving like tumor cells? Is the number of KLF cells due to increased specification or increased proliferation, or both?
Those are great questions. So those are very much the questions we're trying to ask now. So I would say that's, I would say I don't have a good answer for you because we really need to quantify everything very carefully. And just, so I think those are all ongoing experiments that we need to do. What I will say is, yeah, whether they clearly, they're blocked way before these cells would go into meiosis. I will say that. So they seem to be they seem to be blocked at an even earlier stage based on the few markers that we've been looking at, but we still have to do very careful quantification to ask, has the proliferation index changed within those cells? I'm going to stick with hyperplasia at the moment rather than tumor, just because we don't really have evidence that they're behaving like tumors, but there's a lot we need to do to answer your very precise and thoughtful questions.
Great, thanks. Blanche is asking, Blanche Capel, Phil, you mentioned that the nanos-positive germ cells are present in asexuals, but they don't differentiate. Do the ovaries and testes form in asexuals, but also fail to differentiate?
Yeah, so great question, Blanche. So the ovaries, they basically, so the asexuals form primordial gonads that have, we know a lot more about the testes just because we've had the markers for so much longer, but I can tell you that even in the asexual planarians, that DMD, the DMRT homolog that we call DMD1, which is expressed in the somatic gonadal cells, those cells are present in the asexuals, and those cells are required for regenerating the nanos positive germ cells. So the asexuals are able to make germ cells. If we knock down nanos in asexuals, those germ cells are lost. But in both, the somatic gonad seems to be there in both cases. And I'm sorry, I've lost my train of thought. I think I answered everything. Was there some,
I think that's good. We've still got a couple more questions and we're almost out of time. Joni is asking, have you considered treating with serotonin reuptake inhibitors to see if you can phenocopy the ADC knockdown?
Yeah, Umair's been playing with that. I don't think we've had any luck just yet.
Okay. And then Gary Wessel is coming back with asking a detail. Is PKS, you mentioned, polyketide synthase? What might it be doing in gonads?
Yeah, Gary, that's a great question. So basically what I should tell, so PKS does not, so we called it PKS based on a historical, based on its identification in a Planarian paper. So what I should say about that gene, which is very interesting. So the first thing is, it is not polyketide synthase by any stretch of the imagination. So somehow it got that name based on probably a very low hit, probably a non-significant hit with this particular sequence. But the sequence is really interesting. So it is not polyketide synthase. And we really do need to call it something else. We just haven't come up with a good name yet. And we have to, it requires explanation to do that. The thing that's fascinating about this is that this particular gene was identified from single cell sequencing work from Alejandro Sanchez's lab. So Alejandro, Alejandro's lab, along with several others, has been performing single cell sequencing analysis on the neoblasts to characterize their heterogeneity. And they hope to use that to define the pluripotent subset of neoblasts. And they were able to identify a cluster that they could sort with a cell surface marker called T-span1. And so using antibodies against this T-span1 that was enriched in this particular cluster, they could see enrichment of that clonogenic neoblast rescuing activity, that subset of neoblasts that rescues when you introduce into lethally irradiated worms. As part of that, one of the genes expressed in that, detected in that cluster, was this gene that they called PKS1. So what's really interesting about this gene is that we actually can't detect expression of it in uninjured animals. So in uninjured animals, in both sexuals and asexuals, we see it as a marker of these very early, as a female germ cell marker. However, and in fact, I might have it on one of my next slides. Is it my next slide? I'm not going to show it. Basically, what PKS1 winds up being upregulated in neoplasts upon amputation. So it seems, so there's this really interesting parallel between the neoblasts that are responding to wounding and now the expression of female germ cell specific genes, as indicated by at least PKS1, which we only see in the female germ cells in uninjured animals. So that's a long answer to say that this is probably not the gene that you're interested in, but we think it's interesting anyway, because I think it shows this really intriguing link between the neoblasts that are responding to wounds and the female germ cells, and perhaps that pluripotency program showing up during wounding. And also perhaps showing up when we dissociate planarians and remove the cells from their native environment. They might also express this gene. So.
Great. Gary also had a follow-up to that, which was, what's the evidence that the nanos positive cells in the asexual animals are actually germ cells?
Yeah, I would say really the only evidence we have is that they express nanos and these other germ cell markers, and we find them associated with these DMD positive cells. And so it's basically, I would say, just based on the molecular markers that we can associate with germ cell development that are distinct from the neoblasts.
I have to assume that the studies of the developing gonad from the little planaria are much harder than the regenerating planaria, but you're going to get a great tool set now to be able to compare those. Do you know much about that?
Yeah, so that's one of the things that we're actively doing. And we really, yeah, so I would say we don't know much now. Planarians are terrible, they're terribly non-prolific as sexual reproducers. They're really good at regenerating, but they're really lousy at laying eggs that are that have lots of planarians developing, especially this species. This species is really not good at it. So our colony is finally laying, like we have lots and lots of hatchlings now, and these experiments are now becoming totally doable. So we'll be doing a lot more of that as we move forward. But it will be really intriguing to ask what are the parallels and differences between just post-embryonic development and regeneration? And so all very much ongoing questions.
Wonderful. Thank you so much. And thank you everybody for, we just have one last question. I guess I'll try to sneak that in there. Peter Nichols is saying, beautiful studies. Do all gametes in the sexual and early germ cells and asexual planaria arise from neoblasts? And if so, do you view neoblasts as pluripotent cells of the germline, like a blastomere in mammals? Or in other words, is this reprogramming of somatic cells or differentiation of neoblasts to germ cells?
So I think it's very much it's differentiation of neoblasts to germ cells. So neoblasts are actually Alan Spradling used to just say neoblasts are just germ cells. So I think, so there's clearly aspects about neoblasts where they look very much like germ cells based on many conserved germ cell factors, but there clearly is also in this germ cell lineage something different. So we see expression of KLF4 and nanos, which we don't see in the main neoblast population. And we also know that when we knock those genes down, we can lose the germline entirely without affecting any of the somatic tissues. So they're not required, whereas if you knock down the genes that are required in the neoblast proper, like peewees, basically you completely lose the ability to regenerate and you destroy the neoblast population. So this is really a stage in differentiation of neoblasts as far as I would say.
Okay, great. I think we managed to catch everyone's questions. Thank you so much. I will give this back to Saima because she will have a poll here for you guys.
Yes, thank you. So the poll is now on your screen. We just wanted to get some quick feedback. It is anonymous, so please fill that out before you leave. And thank you so much for your time and attention today.
Thanks, everybody.
Thank you. I just wanted to say the recording will be available on the SSR website once we've made sure that we have everything just cleaned up the edges of it, but it will be available online. All right, thank you, everybody. I think it's just us now. So thanks so much,
Okay.
Phil. Thank you so much, Erica. Have a great night.
You're muted, Erica, but.
Oh, can you see the poll? Okay, Phil, you did great.
Oh, good. I did.
Yeah.
Great.
Okay.
Great.
Excellent. All right. Well, thanks so much. That was really fun.
Oh, thank you. That was amazing. So beautiful. Yeah.
Thanks.
The only bummer for me was since I had to read all the questions, I couldn't really listen to your answers because I had to, focus on the questions. So.
Yeah, probably not a bummer. So.
No, really nice. Okay. Yeah.
Great. Okay.
Thank you.
Well, thanks so much.
Bye-bye.
Okay, we'll see you. Okay, take care, Erica.
Say hi to your
care,
family.
You too. Okay, bye-bye.
Bye-bye.